So, welcome everyone to our virtual seminar. And first of all, I would like to thank Professor De Jong Yao and also Professor Valdez Souza for organizing this virtual conference on the 20th World Congress of Psychophysiology. Our symposium will feature particularly the topic of training the brain of real-time neurofeedback approaches, which has been developed during the last 10 years and which has become more and more major. So from being a very experimental tool, we now see a strong methodological progress for building this together with multivariate pattern analysis, also with connectivity measures. So moving into a more methodological direction, training more holistic measures of the brain and maybe more suitable measures. And on the other hand, we see that the great clinical promise that real-time neurofeedback maybe has to modulate brain regions in patients with brain disorders is becoming more and more reality. So there are initial clinical trials that either can show or have proven strong clinical potential. Therefore, I'm very happy and excited today to lead a symposium on this and also to have very exciting speakers here from all over the world. So we'll start with Aurelio Cortese from Japan. It's then followed by Michael Leurs from the Netherlands, followed by myself from currently residing in China, and followed by Jana Zwerings and also Professor Klaus Messiek, both from Aachen University. So let's give a quick outlook what we will be talking about. The first talk will be held by Aurelio Cortese. He will talk about decoding neurofeedback, about process-based mechanistic neural targeting. The second talk will be held by Mike from Maastricht University, and Dr. Leurs will talk about semantic neurofeedback and also particularly about representational similarity neurofeedback. The talk will be followed by my own talk, where I present a novel connectivity-based neurofeedback and translation of this to FNIRS, which may open more clinical applications, which will directly lead over to more clinically oriented talks. For instance, the talk from Dr. Zwerings from Aachen, from the Aachen University, and she will talk about real-time neurofeedback solutions for symptom-specific treatment of psychiatric disorders, including different disorders like trauma-based disorders. Then, at the final speaker, we will. I'm very happy to have Klaus Messiek on the and Dr. Messiek from the Aachen University will specifically talk about the learning process during the neurofeedback and how we can optimize this. So basically from operant conditioning to social hyperscanning. And I'm now very much looking forward to all of the individual talks. Thanks again to all speakers, and I hope you enjoy the talks.
Thank you, Ben, for bringing us all here. This is a wonderful symposium that you have organized, and I'm very happy to have the opportunity to give this talk. Today, we'll be talking about our recent progress with a special type of neurofeedback that allows fine-grained targeting or manipulation of neural representations or psychological processes. There are multiple driving goals behind the development of the decoded neurofeedback approach, but one of them is clearly the ambition to find new solutions to a growing problem in our societies, mental illnesses, from anxiety, stress, and phobia to depression and OCD. These are all often illnesses that are refractory to standard treatments. And so the central tool at the basis of decoded neurofeedback is combining functional magnetic resonance imaging with machine learning. The basic idea is we let a machine learning algorithm learn how brain activity patterns are related to some behavioral variable or physical stimuli in a supervised manner. So the algorithm will learn eventually that some of the voxels that we recorded with fMRI are important to distinguish the nature of the stimuli. And so those voxels will have large weights and then can be used later on for prediction or for further analysis. So here, why, you may wonder, why mechanistic neural targeting? So why are you talking about mechanistic neural targeting? The reason is really intrinsic to the machine learning decoding approach. It has a high specificity because it is centered around information content and because it is based on localized spatial temporal activity patterns. So as an example, you can imagine that, say, in the early visual cortex, you have an average fMRI signal when viewing green or red is about the same, both are colors. But if we look simply at the activity level of this region, then the two conditions are probably indistinguishable. Yet, if we look at the pattern of activity, then they should be quite different and should allow us to know what the participant was actually viewing, green or red. We can go well beyond, we're not restricted to these simple visual features. So more complex objects or behavior variable or even abstract constructs can be used to build a decoder. So once we have a decoder, we basically have our central ingredient for decoded neurofeedback. So briefly, in decoded neurofeedback, we built a decoder to classify neural representations of two or more conditions. So for example, red versus green. Then, usually a couple of weeks later, we use the same decoder in a real-time setting. It's a kind of in this cartoon depiction. So essentially, we measure brain activity with fMRI. We extract the moment-to-moment activity patterns from the same area, same voxels that our decoder was trained on. And we input these activity patterns into our decoder. So the decoder would then calculate the likelihood that this information that is represented in this area at this point in time corresponds to our target. For example, here, red color. So this likelihood is immediately translated by the decoder and by our program into a visual feedback. For example, the size of a disk, as we commonly use in our experiment. So here the participants usually only told that the task is a game they are playing, the goal of which is to magnify the size of the disk in order to obtain larger monetary reward. What they're not told about is that the size of this disk really corresponds to the likelihood of the decoder that is detecting a target representation in the neural activity. So what really happens is that we are reinforcing the neural activity with monetary rewards. So with time, the brain learns to associate a pattern of activity with a reward. And this is extremely useful and powerful, as I will show you in the next slides. So let me start with actually potential clinical application of this neurofeedback approach, because this is the one that I think has very strong opportunities. So I'll introduce two proof of concept studies that we developed to tackle fear and traumatic memories, as well as phobias. So why develop such treatments? So one of the big issues in treating fear-related disorders is the high dropout rate. So for many patients, it is difficult to go through exposure therapy, where one is presented with the fear objects. And so it would be beneficial if we could develop an approach that is able to reduce fear responses directly in the brain and therefore non-invasively and with little distress. So we first aim to reduce fear responses to noxious stimuli by targeting representations in the visual cortex. So in a classic fear conditioning paradigm, participants learned that some stimuli, the CS plus here, were predictive of an electric shock. Because the stimuli here are differed by very simple visual attributes such as color, we can build a decoder in visual cortex to identify the specific activation pattern of each CS, and color can be relatively easily decoded. So you can see that the initial physiological response to fear responses measured both at the skin conductance as well as the amygdala reactivity were identical across the two CS pluses, target and control. So what we next did, we used decoded neurofeedback to target the neural representation in visual cortex of the CS plus. Participant did not know which one was the targeted, what was the goal of this procedure, but what we did is that we associated rewards with the neural occurrence of the target CS plus. So you can see this as a kind of counter conditioning procedure, since those noxious stimuli are now associated with something more positive, a reward. And after training, we measured again the physiological fear response to those stimuli. And crucially, we saw that only the responses to the target CS plus was reduced. So the fear response remained high for the control, but was reduced for the target CS plus. And this was a great result, it seems that we can reduce fear directly in the brain. Nevertheless, in truth, this study suffered from two limitations. So first of all, color gratings are not necessarily scary in the real world. They're not really naturalistic stimuli. And so it remains to be tested whether this approach is effective also with actual stimuli that people can be scared of, snakes, spiders, you name them. And second, more importantly, actually, in order to get the pattern for the target stimulus, we had to construct a decoder for each individual. We had to show those stimuli to participants, which basically brings us back to square one. Luckily, Huxley and colleagues, they developed a way to bypass this issue. So this was a study that was led by Vincent at ATR, which I was involved as well. And we went on to replicate this initial reduction of fear for common animals and objects. So in a hyperalignment, one constructs a common space from the fMRI data of several participants. So such an abstract space is created by simply applying a set of linear transformation to the original fMRI data, iteratively and across participants. This approach results in a transformation matrix that allows us to go from the individual space, voxel space, to this common space, and then back. More specifically, this means that we can now infer a target activity pattern, for example, of snakes, in our designated participant, but from the activity patterns of a group of surrogate participants. So this means that we don't even need to ever present the image, as long as we have other images that were presented to all the other participants, these other object animals. So to replicate the previous studies, we added three important modifications. First of all, we used 40 different categories of common objects and animals, of which humans are often afraid. We bypassed the need to show these images directly by using hyperalignment. And the target and the control images were selected as two for which the participants were the most afraid of, but the decision of assigning the target and the control conditions was defined by the program. So effectively, we had a double-blind, placebo-controlled design. So we used decoded neurofeedback to associate the reward with the neural representation of the target category. And we showed that again, after the training, we have reduced response, both in terms of amygdala activity and in terms of SCR, only to the target condition. The control condition shows no difference in before, pre, and post. So this means that we can now actually use this as a potential treatments, and my colleagues in Japan and the US are running trials with subclinical populations. So I've showed you here two decoded neurofeedback studies, but we have actually run multiple such studies at the ATR and targeting very different brain regions and very different stimuli or psychological construct. So effectively, if we consider that we are able to construct a reliable decoder in a given area, we can theoretically do a decoded neurofeedback study. So this really lends to the idea that it is a very general approach. But what is the mechanism that enables decoded neurofeedback effects? So first of all, we now have strong evidence that it is based on reinforcement learning processes. It comes from both from a meta-analysis and simulations. We know that there is a relatively close mapping between the measured voxel activities and the underlying neural activity, but it remains unclear how the brain can actually learn which area is being targeted in decoded neurofeedback. And so this leads us to another question. Can we test the ability of the brain to explicitly learn an arbitrary neural boundary, which is essentially what we do with decoding and neurofeedback? And answering this question will help us better understand decoded neurofeedback because it will provide proof that the brain can efficiently learn an arbitrary boundary in neural space through trial and error. So we designed an experiment in which we asked participants to bet on the content of their own brain activity. So they did not know this. We told them that they had to learn to choose between two options to obtain as many rewards as possible. So choosing, let's say, option A and option B. We used a decoder to determine the likelihood of an internal mental representation, let's say R or L, and either in visual cortex or in the prefrontal cortex. And this was based on the fluctuation of spontaneous activity. Now, when the decoder detects state R, the optimal action should be A. When the decoder detects mental state L, the optimal action should be B. This is a formidable problem because there's almost like an infinite number of mental states, and the brain has to learn this kind of boundary between those that defines those two sort of arbitrary R and L states, and then associate this with an action to obtain rewards in a non-conscious way. So did participants learn the association at all? Bear in mind that they were not informed about the fact that decoding determined whether their choice were optimal or not. So here we plot the probability of making an optimal choice, A or B, depending on the brain state and the trial. And although this effect was small, we see that participants seem to learn. They can learn this association. And in session 3, we have a control condition. So we translate the decoding output as a visual stimulus, and we see that they can consciously learn the association. But what is what drives the learning? Well, one hypothesis is that confidence and reinforcement learning can interact to make this problem less complex. And here we have results from a modeling. Basically, we use reinforcement learning, and we investigate if there is a link at the trial by trial fluctuation between prediction errors, which are null mark of learning, and those confidence rating that participants were giving. We see that as learning progressed, this relationship between confidence and reinforcement learning becomes increasingly stronger. So namely on trials in which participants were very confident, they also had lower prediction errors, meaning there was more certainty in their choices and more certainty in the actual learning. Because confidence happened earlier in the trial than the actual outcome was before the outcome, this means that confidence is inherently linked with learning. We went next to show this at the neural level, and we show again that. So what we did, we built a decoder in dorsolateral prefrontal cortex for confidence, high versus low, and a decoder in basal ganglia for high versus low prediction errors. And then we pair the output of those decoders and measure the degree of independence. If they're independent, they're not, those two areas are uncoupled. If there is dependency, then it means that we have a coupling. And we see that with learning again, across the session, across higher performances, we have a stronger and stronger coupling between those two areas. Now here, it's important to notice that this coupling is, we're not talking only about a generical neural coupling between those two areas, but really about a specific coupling that is related to confidence and reinforcement learning. So to summarize this study, we show that an arbitrary boundary can be learned within a limited number of trials. This amounts to participants being able to read and use their own brain states to get rewards. Given the strong relationship between confidence and reinforcement learning, this maybe points to the fact that confidence and metacognition could be a means to accessing internal non-conscious information, and opens question for future work, which is whether metacognitive abilities are important for neurofeedback learning. So it's a good point now to summarize also the key properties of targeted decoding neurofeedback. It has high specificity and can be run without participants' awareness. Paired with hyperalignment, the approach offers the possibility to avoid presenting the distressing stimuli altogether, so to construct a decoder. It can be run in a double-blind placebo-controlled fashion. And finally, because it taps into the reinforcement learning process, it can be implicit and efficient. But we can expand on the real-time framework. Instead, for example, associating neural representation with rewards, we can instead monitor brain activity fluctuations to detect the occurrence or modulate task requirements for example, to determine what is the best timing for a stimulus presentation. Of course, there are limitations to this approach, and these are first of all that we need a good decodability of the target information. We don't have control over the reinforcement learning process because it is passive from the experimental point of view. So future work will have to investigate what are the markers of successful learners prior to the training. And finally, what happens if the brain reinforcement learning machinery is dysfunctional, as is the case in schizophrenia or depression. But do we have only decoded neurofeedback? Although I've talked about this, again, we can actually use the connection between areas and use machine learning to find specific network connections that can be targeted with connectivity neurofeedback. So all in all, I would like then to thank really important collaborators that have been taking part in these studies, funding agencies, and of course, most of all, you all who have watched this talk for your interest. And I look forward to hearing your questions and thoughts.
Welcome, everybody. My name is Ben Becker from the University of Electronic Science and Technology of China, and I will today present a novel neurofeedback training that we evaluated. So basically, previous fMRI-based neurofeedback trainings primarily targeted regional activity, so trained subject to control regional activity. And in further development of these trainings, we aimed to train subjects to gain control over pathways. And we specifically targeted pathways connecting the amygdala with lateral prefrontal regions, because from previous studies we know that these regions, or this connectivity between these regions, is involved in emotion regulation, anxiety regulation. And this, although is the background of our studies, so we are trying to improve emotion regulation, although particularly in clinical context. And therefore, in the next steps, we further translated this technically rather challenging connectivity training to a maybe clinically more applicable training that uses FNES. And as already mentioned, our background is mainly a clinical background, and we want to improve emotion regulation and anxiety regulation because these things are disrupted across most mental disorders. So we see exaggerated anxiety and deficits or impairments in controlling negative emotions in general, or anxiety in particular, across several disorders. Most pronounced, this is obviously in anxiety disorders, but also in trauma-related disorders or addictive disorders. We see these deficits. Currently, these deficits are treated either pharmacologically or with behavioral cognitive therapy, and these interventions have some success. However, they don't work optimally for all patients, and with several of these measures, the efficacy is rather moderate, and lots of the pharmacological agents come with negative side effects. So there's currently a lack of efficient treatments for this. And over the last 20, 30 years, actually no new pharmacological agent improving emotion regulation, decreasing anxiety has reached the market, at least for applications in humans. So on the one hand, we urgently need better and new treatments. On the other hand, we have an increasing evidence or increasing knowledge from fMRI, MRI studies in healthy subjects, but also in patients, that have mapped the corresponding brain systems that are dysregulated in patients with emotion regulation disorders, but that are although underlying healthy, intact emotion regulation. So, for instance, in a big number of patients, we have meanwhile seen that often in disorders characterized by emotion regulation deficits, characterized by strong anxiety, the amygdala is too strong active, and maybe all the other emotion regulation or other emotion reactive regions like the PAG. At the same time, often prefrontal or all the parietal regions that are involved in regulation, in executive functions, in downregulating emotions that have direct connections with the amygdala, activity is too low. So they show decreased engagement. This may underlie emotion regulation deficits. And in more and more studies, we actually see that it's not only the regional activity that is important for these deficits, but it's also particularly the interaction, so the communication between these regions. And for instance, we have seen and shown this, and other groups have shown this, in several disorders that are characterized by emotional dysfunctions. So for instance, a couple of years ago, we could show that reappraisal, so explicit emotion regulation deficits in drug addicts, are mediated by deficits in the connectivity between the DLPFC and the amygdala. So we have seen that the amygdala is still, when these subjects try to downregulate their emotion, is still stronger active than in healthy controls. However, the most critical part was the lack of regulation via the prefrontal region, and particularly via the lateral prefrontal cortex. In addition, and this is important, we and others have conducted treatment studies, so basically assessed patients' neural activity in patients during emotion regulation before and after they underwent successful treatments. For instance, in trauma-related disorders or in stress-related disorders, we have seen when the patients become better over cognitive behavioral therapy or over other behavioral interventions, the connectivity between the prefrontal regions and the amygdala normalizes, so becomes normal again when the symptoms decrease. This is actually a very important finding because this indicates that the amygdala prefrontal regions are connectivity between these regions is treatment sensitive. So basically, it's not like a fixed trait that we can't influence, but if we modulate this, the patients will recover and are better able to regulate their emotions. So this makes these pathways a quite interesting, probably also quite promising treatment target. These circuits models or circuits that we identified patient studies also align very good with recent network level models of emotion regulation. This model, for instance, from Atkin and colleagues, also proposes that during the initial encounter of something negative emotionally, for instance, like a threatening stimulus, the amygdala and all the other regions, like the PAG shown here in red, become quite active in the first moment, but they are downregulated by prefrontal regions shown here in blue. And importantly, different circuits connecting the prefrontal cortex and the limbic system, like the amygdala, are involved in different emotion regulation functions. So, for instance, whereas the VMPFC, so the medial prefrontal regions, and their regulatory control over the amygdala have been involved in implicit emotion regulation, such like fear extinction, implicit emotional conflict processing. The lateral prefrontal regions and their connections with the amygdala have been clearly involved in explicit emotion regulation, so in volitional emotion regulation. So this now basically gives us, together with the patient studies, a relatively clear and promising pathway when we want to improve emotion regulation or decrease, or explicit emotion regulation and decrease, for instance, anxiety. These pathways connecting the lateral prefrontal cortex and the amygdala might be a highly promising target. To now test our hypothesis whether we can directly modulate these pathways and whether this has an effect on anxiety and on emotion regulation, we capitalized on real-time neurofeedback and on current technical developments in the field. So real-time neurofeedback is a relative evaluated technique, meanwhile, for modulating neural activity. And from my perspective, the underlying platforms, all of the underlying technology is relatively elegant. So basically, subjects are displayed neural activity from a specific brain region often in real time. So while the subject is in the scanner, the images are processed in near real time, and the subject is provided with a feedback, an online feedback, on how strong activity in a specific brain region is at a current time. And over this has been shown in several studies, subjects can learn to regulate their brain activity in a specific region. Several studies before were tried to use this for improving emotion regulation by either targeting the amygdala and downregulating the amygdala, this has been shown, or by increasing prefrontal activity. We now wanted to go the next step and see whether we can also train circuits, whether we can actually train the communication between the amygdala and the ventrolateral prefrontal cortex, which we would expect probably has an effect on emotion rate. And we conducted a study, and we wanted to find out, can subjects learn volitional control between the amygdala and the ventrolateral prefrontal cortex when we provide them with online feedback about connectivity in this pathway? And does this also have a clinical measurable effect on their emotions, and particularly on their anxiety? To this end, we conducted a randomized controlled trial where subjects underwent neurofeedback training, and we implemented this training basically also in an emotional assessment. So before the training and after the training, we asked subjects to rate their anxious arousal. And we recruited high anxiety subjects to increase the clinical relevance. After that, subjects underwent 2 normal fMRI paradigms, which served as localizer. So basically, we mapped the ventrolateral prefrontal cortex amygdala pathway with this normal paradigm. And in addition, we mapped the motor pathway. So it was a simple motor task, the second task, where we measured the motor pathways between the left and right motor cortex. So we now had 2 pathways, one involved in emotion regulation, or supposedly to involved in emotion regulation, connecting the amygdala and the ventrolateral prefrontal cortex, whereas the others connect the motor cortices and is likely not involved in emotion regulation. So we expected that when our training works, that the connectivity in the VIPFC amygdala pathway should increase over the 4 training runs, whereas anxiety should be. On the neural level, we actually saw, and this is shown here in the red in the upper part, that over these 4 training runs, the connectivity in the pathway between the amygdala and the VIPFC increased. Importantly, and this is shown in blue on the lower part, the connectivity in the sham group, so who received motor or circuit power feedback, did not change. So this shows that the feedback itself actually is a very important driving factor. On the behavioral level, we observed that only in the session where a subject received the real feedback from the VLPFC amygdala pathway, anxiety decreased. So we now have a training that can be used to train subjects to gain control over a pathway that is importantly involved in emotion regulation. And this also has the potential to decrease anxiety. So this has a quite high clinical potential. However, on the other hand, if we're now thinking very pragmatically and we want to move with this into the clinic, we have several technical hurdles to overcome. So in general, it's a rather technically demanding and also relatively expensive technology. So, for instance, although in the clinical daily practice, the MRI systems are highly occupied, you need trained technical assistants. They come at very high costs per scanning hour, it's movement sensitive, and it's maybe not the best environment for patients with anxiety or stress being trained, because it's often from patient experiences is pretty it's pretty stressful being the fMRI. So therefore we now are considering, can we translate these training actually, for instance, to a more simpler technology. That is, for instance, FNIRS, which is an optical imaging technology, which is relatively robust against movement, which basically works with caps, similar like EEG caps, and also uses optical signals to acquire bold changes in the brain. However, so it has robust advantages, and it's also quite more cost efficient as fMRI. However, it comes at the cost of the limitation. This is a limited penetration depth. So the penetration depth into the skull, it can basically just reach the cortical surface. So we now are thinking, okay, we can do this, but we can just focus on the ventrolateral and prefrontal cortical region. So what we did, we first mapped the region that we trained as one node in the pathway that we trained, the DLPFC. We mapped it using fMRI on the FNIRS electrode, and we could basically identify a channel that is very much aligned with the DLPFC region that we trained. We next implemented the FNIRS into a real-time neurofeedback platform, and basically which works very similar to the fMRI neurofeedback platform, just that it uses the FNIRS continuous signal, and the continuous signal is displayed to the subject in real time. To now test the general feasibility of this training and to also test behavioral effects, we conducted an initial study where subjects underwent 4 training runs of FNIRS-based neurofeedback of the DLPFC. We could see that over the 4 training runs here shown on the left, the subjects that received feedback from the DLPFC were able to increase DLPFC activity over the 4 runs. In contrast, here shown in the middle, the sham group did not show any changes, which again is important because this indicates that actually the neurofeedback signal itself is an important driver. It's not just increasing concentration or increasing unspecific effects. We also saw some, but rather more subtle effects on the behavioral level, so implicit executive control task, and there we saw that the reaction time decreased slightly in the group that received the real feedback as compared to the sham control. Our aim was now in the currently ongoing study to basically close the circle, to see or to find out and to test whether if we train the DLPFC via FNIRS neurofeedback, do we also change connectivity of the amygdala, which was actually our starting point. And this is now data from my study, and I present here data from, I think, around about 32 subjects in the between subject design. So briefly, subjects were randomized to either an FNIRS feedback from the BLPFC or a sham Yoki condition. They underwent 4 runs of this training, and subsequently we went into the fMRI scanner, where we assessed, among others, resting state fMRI. And our initial preliminary analysis indicated that the group who received the VIPFC neurofeedback and who basically increased activity in this region over the 4 training runs showed decreased intrinsic activity of the amygdala. And on the network level, I'm showing you on the right, which is a functional connectivity analysis, we could see that connectivity of the amygdala with regions of the frontoparietal control network increased in the group that received the VLPFC training as compared to the sham group. So maybe this now allows us to close the circle and to say, okay, if we now train just the VLPFC as call node, we can in turn affect the lower cortical or subcortical circuits again. Connectivity-based neurofeedback is possible, so subjects can learn control over pathways using fMRI neurofeedback. If we target pathways that are specifically involved in emotion regulation or anxiety regulation, this also has a behavioral effect, so it can decrease anxiety, at least in our study. And we can then transmit this region, the VLPFC region, to a maybe more robust, more clinically utility platform like fNIRS. And there we see that although this is feasible, subjects can learn to increase activity in this region using fNIRS, which in turn then again affects the underlying amygdala prefrontal regulatory circuits. So maybe this is one step into the clinic now. Okay, thank you. Obviously, this is not all my work, but I have lots of help from my team in Chengdu and from my collaborators in China, Germany, and all in the Netherlands. We are currently also looking for PhD and postdocs. If you are interested in our topics, just
Welcome to my talk. My name is Jana Zwerings. I am happy to be part of the symposium today, and I would like to present to you some of the projects that I was working on in Klaus Mathiak's research lab during the past couple of years. My research activities are mainly focused on the investigation of dysfunctional cortical networks and the development of real-time fMRI neurofeedback paradigms in the context of mental disorders. And this is also what I will be talking about mainly today. So in the next 15 minutes, I want to introduce to you some of the projects in our research group that reflect the diversity of approaches that can be taken within the context of neurofeedback paradigms, but also the different symptom clusters that can be targeted. And to give you an overview, we were working on projects in patients with depression, trauma-related disorders, schizophrenia, and recently also substance use disorders. And my main focus will be on trauma-related disorders today, where we predominantly were working with or to support prefrontal control mechanisms, for example, to enhance emotion regulation. But I will also introduce to you one project in patients with schizophrenia with regard to auditory verbal hallucinations and feedback of the left hemispheric language network, and a network-based approach that we took or are recently taking to enhance impulse control in substance use disorder. So next to these different symptom clusters and clinical disorders, a focus of the research group has also been to develop different feedback modalities and paradigms to enhance learning during neurofeedback. And here you can see one of the paradigms where we use an explicit emotion regulation strategy combined with these negative images and simple numeric feedback. But we also have, for example, social feedback and immersive virtual environment paradigms that we're working with during the past 2 to 3 years. As I said, I want to start with trauma-related disorders and more specifically with post-traumatic stress disorder. Post-traumatic stress disorder is frequently associated with hypoactivation in frontal areas, and this includes also the anterior cingulate cortex. And the hypoactivation in the ACC seems to be specific to individuals who develop the disorder compared to those that experienced a trauma but did not develop PTSD. Furthermore, a longitudinal study in a Canadian research group showed that ACC activation or increased ACC activation after psychotherapy was associated with symptom improvement. And based on these studies, we were thinking and decided to try to directly target the ACC with neurofeedback and see whether patients can learn to regulate this area. And we decided to use a social feedback paradigm that you can see down here. And social feedback means that during the regulation phases, the smile of the avatar changed. So the better the regulation, the wider the smile. And this is quite rewarding if you imagine someone smiling to you upon regulation success. So what we did find was that actually both groups, so the healthy controls and the PTSD patients, managed to regulate portions of the ACC. However, this effect was much more pronounced in the control group, as you can see in this group comparison here for anterior and posterior cingulate cortex regions. A linear regression analysis also confirmed this observation, indicating that there was a higher learning rate in the controls compared to the PTSD patients and portions of the ACC. The PTSD patients showed a higher activation in the frontal medial cluster, and we thought that this may reflect compensatory mechanisms during regulation or to be able to achieve some regulation. Here you can see that, symptom severity also predicted ACC regulation success, and taken together, these results may indicate that a dysfunctional ACC regulation contributes to the etiology of PTSD. Interestingly, after the training at the follow-up, we also saw that there was a reduction in symptom scores. However, this has to be interpreted with some caution because we didn't have a control group here. So it can also be spontaneous remission or similar. In our second project in patients with PTSD, we took a little bit different approach. So as I already said, in PTSD, there's often hypoactivation of prefrontal control regions that can be observed, but also in response to negative images, for example, a hyperactive limbic system. And cognitive reappraisal is one strategy from cognitive behavior therapy that may counteract this activation pattern because it has been associated with increased activation in frontal areas. And we were now trying to find out with this study whether neurofeedback can help us to boost cognitive reappraisal in patients with PTSD. And we wanted to see whether this has a positive effect on emotion regulation, but also a symptom severity. And we used the paradigm that you can see down here. And the participants saw these negative images and just had to look at them during the view phases, or they had to actively reevaluate the content of the images in the reappraisal phases. Then they received feedback from our target region in the left lateral prefrontal cortex. And this number reflected the bold response in this region during the reappraisal phase compared to the view phase. Now, what did we see? Indeed, we observed reduced activation of the amygdala during the neurofeedback compared to the control condition. However, for the left inferior frontal gyrus, our target region, we did not observe the expected effect. So here, activation was lower during neurofeedback. Interestingly, and that was quite important for us, the amygdala responses during the neurofeedback training, but not during the control condition, were associated with symptom improvement 4 weeks after the training. And the same pattern also emerged for a negative effect. But we didn't want to stop at this point, and we were also looking at possible changes in resting state connectivity after the training. As you can see here, our analysis showed that there were selective changes after the neurofeedback compared to the control condition in connectivity between the left inferior frontal gyrus and the bilateral amygdala next to some other regions. And our region-to-region analysis confirmed this result, but showed that the effect was particularly true for the right amygdala and the superficial and basolateral portions of the right amygdala. We also wanted to see whether there was like an association between emotions or emotional ratings and symptom severity with connectivity. And we saw that bilateral amygdala connectivity with regions in the parietal lobe predicted changes in positive effect, but not for negative effect or symptom severity. So while these results are encouraging, of course, there are some open questions left. And this is, I guess, a good way to introduce our last project in trauma-related disorders that we are currently working on. Here, we integrate neurofeedback to support resilience in traumatized refugees. And more specifically in Arab refugees, we have a randomized double-blind placebo-controlled design. So here we really want to pin down neurofeedback-specific effects. We have this active or this target region, again, in prefrontal areas. But we also have an active control region and a non-neurofeedback emotion regulation training condition. We want to compare outcomes, or our primary outcome is resilience. Here we decided to use resilience because it is a protective factor, particularly in the context of mental disorders. And since refugees are often confronted with post-migration stressors in everyday life, we wanted to see whether we can boost resilience, whether we can have a positive effect here with the training. And for this, we have a follow-up after 1 and after 8 weeks to have a first idea of an association between this training and resilience. We created a novel immersive virtual environment feedback modality just for this study. And as you can see here, based on the brain regulation or based on the regulation success, the environment changes. So it gets brighter and there are more flowers that are blooming. Also, it is very dynamical. So there's a nice breeze going through the trees and the meadow. And sometimes there are some birds flying to make it immersive. So with this, I would like to come to our study targeting auditory verbal hallucinations and schizophrenia. We were focusing on auditory verbal hallucinations because it is one of the most common symptoms for schizophrenia, and it has been associated with functional alterations in language processing areas. Furthermore, there are some studies that showed that there are changes in connectivity between frontotemporal language network and also the default mode network. So we created a study where we target the real-time fMRI neurofeedback study, where we target the left hemispheric language network. And this means the left inferior frontal gyrus as speech production area and the left posterior superior temporal gyrus as speech perception area. These were defined in each individual in a functional localizer. And then we had 2 days of neurofeedback training. One was an upregulation neurofeedback day and the other downregulation neurofeedback day. This was randomized and double-blind. And after 1 month, we conducted a follow-up Also, again, before and after each day, we had a resting state measurement to look at the changes in functional connectivity after the training. So during the training, what we did see was that indeed we have, during the upregulation compared to the downregulation, higher activation in the posterior superior temporal gyrus target region, also comprising like some wider cluster bilaterally. And we also did see that across the sessions, the probability to achieve higher feedback values increased. That was true for both groups, patients and controls, and also for both conditions, up and down regulation. We were also asking the patients whether they had the impression to be able to control the feedback signal, and a high percentage of about 70 to 80% indicated that this was the case. There were also changes in the severity of auditory verbal hallucination ratings after the neurofeedback training, but these were not associated with regulation success. Looking at the changes in resting state connectivity, we observed a selective effect of the downregulation compared to the upregulation condition between the pooled seat region, so the left inferior frontal gyrus and the left posterior superior temporal gyrus, and nodes of the default mode network, as for example, the parietal lobes regions. In the group comparison, now just for downregulation, we saw that there are also differences between groups in this connectivity. And when we were further looking into this, we saw that this effect is mainly driven by the patient group. So the patients had higher connectivity after downregulation between the left hemispheric language nodes and these nodes of the default mode network. And this is quite interesting because based on the literature, it may be indicating that there's a better discrimination between externally and verbally and internally generated verbal material. The connectivity between the left IFG and also nodes of the default mode network was also associated with symptom severity. So higher connectivity was found in those individuals who showed lower symptoms according to the PANS. With this, I want to quickly, just for a minute, introduce the last project that is focused on impulse control and addiction. And I think this project is interesting to mention because we again follow a different approach here. And what we decided to do was we first wanted to conduct a meta-analysis to define the network that we wanted to train with our neurofeedback paradigm. So we conducted a meta-analysis to identify altered resting state functional connectivity patterns in substance use disorder. And this was also done in collaboration with Ben Becker. And here we see that results indicate a hypoconnectivity within a network that you can see here on the right side, comprising the insula, the anterior cingulate cortex, and the striatum. And this finding was kind of interesting because when we were looking in the literature, we saw that decreased resting state functional connectivity in this network is linked to increased smoking behavior and also unsuccessful smoking cessation. And as I said, we are interested in impulse control. So these regions are also involved in inhibiting prepotent responses. So we were thinking, okay, we want to see whether in our case in smokers, if they are able to regulate this network. And then we also wanted to evaluate the behavioral relevance for smoking behavior, but also for inhibitory control by a stop signal task that we are using before and after the training. With this, I want to come to an end. Of course, I want to thank all the collaborators and the whole experimental behavior psychobiology lab. As you can see, there are a lot of things that we have to consider when we are creating these experiments, and this is only possible by having good collaborations and an interdisciplinary team. And of course, I also want to thank you for your attention, and I'm looking forward to your questions.
Good morning, good afternoon, good evening, wherever you feel home in this world. So after having seen already some nice introduction and ideas for clinical application, I will focus a little bit on the type of brain-computer interface and show how we're trying to connect fMRI neurofeedback with social interaction in virtual environments. I don't know how communication will work here from the conference, so I put my email here on the slide, and probably you can also see them in the program. And please don't be hesitant to just email me also if you have some question. We can't discuss it directly here on the conference. So to see how the page change is working. So yes, first of all, I wanted to quickly reintroduce our work on brain imaging during this type of semi-natural behavior, and how we use this as a BCI for studying ecological valid behavior. And then take the next step, how we can use this environment for neuromodulation, and then introduce it into hyperscanning neurofeedback situation. The latter is more a vision, what we are piloting currently. So I won't have much data on this one. Go ahead. So this is data we have already a while ago, where we see if we use virtual environments with social interaction situation. Here, this is a computer game, a first-person shooter, with the advantage that even at those times they had a rather naturalistic display of this not so common interaction. And advantage about these computer games is also that this is freely behaving situations, but in a very circumscribed context. So we can define a specific situation, for instance, something we call active fighting. You see here this interaction where which is finally the aim of the game, but there are also different constructs, for example, being safe. And what was surprising for us, that there were very clear pattern of neural network interactions we could identify using this relatively simplistic model to study semi-free behavior. And this can be applied, as you've seen, to violence, but also something like flow experience. So there's a mixture between something to be not enough challenging and too much challenging. Very important construct, for example, in sports, to get to the right level. And there you see that basically the whole motor system is involved in particularly those situations where flow is experienced. Yeah, and what is important, of course, also for neurofeedback, also we can determine reward areas, reward associated with actions, or reward associated with reward consumption. Yeah, to put that to a more practical level, we were also looking to apply this into more real-life situations. So we're using, for instance, driving simulation together with a motor company. And also here we can see that specific situations, such as alerting to upcoming situation, can be identified in terms of neural networks involved, or to and see that this is of course in the insula, for instance, in the formatio reticularis, and this we can actually use to create fingerprints, and associate that with the remaining psychophysiological responses, which then can be measured also during driving. So we can use it to predict response accuracy and reaction time, which is then potentially interesting for application in driving assistance systems. Also, of course, we can use it to modify rewarding behavior. So, for instance, such a virtual environment where we change visibility from low to high visibility. Of course, in the high visibility situation, we have a higher likelihood for positive rewards, leading us into the situation that we indeed can do operant condition for specific brain signals, as in neurofeedback, or even more straightforward, we can use social interaction environment. Here, the famous cyberball game, where we can see when the other are playing, we are excluded, and when we are included, we perceive this as a rewarding system. And here we can change the reward probability depending on the local brain activity, and thereby reinforce certain networks. Yeah, so the whole system looks like this. There's this virtual environment, could be a VR game. As long as we have control over the environmental logic, we can implement a feedback, and of course also a complete logging system, so we can directly extract behavior measures like reaction times, and thereby see if these interventions have the desired behavioral effect, for instance, to increase responses in patients with depression, to improve negative symptoms, lack of motivation, for instance. Yeah, and therefore we do measure during this gaming the brain activity, which turns out to be rather relevant, to have a very high contrast to noise ratio, to have good signals. So we're using here this now single shot multi-echo, multi-band imaging, and then we need a processing which is also robust of outlier. Here we rely on the Bayesian system, and thereby we obtain signals which are tracking these networks or network connectivity, and can use it exactly to these game engine modifiers, which are having an effect on the reward probability, and thereby induce operant conditioning learning situation. We can first of all show, of course, that we can have a learning effect on the behavior level. For instance, we see how well the control ability is perceived, but also to address anhedonia symptoms, for instance, in depression, we can see that this scales are improved, and often even specifically when we target, for example, the SMA as a network, which is not much directly involved in affective processing, that we see an improvement at its level. Yeah. Yeah. Interestingly, of course, this is a network effect. If we regulate in this environment the SMA, we see that the changes are directly spreading out to navigation areas like hippocampus. Yeah. So to pull this whole thing together, our concept is now to use this social reward and neurofeedback situation, implement them into this virtual environment where social interactions play a core role, and use now between two subjects during hyperscanning the intersubject brain synchronization to obtain increase of similarity of the brain reaction partners. So the idea is here, if we have, for example, patients again with aberrant brain activation pattern, which has been shown for several dysfunctions, we can give them a trainer who's showing the right brain activity, and by increase of intersubject covariance, the network activation is normalized. Yeah, indeed, looking back into the whole hyperscanning literature, the ISC was basically one of the first parameters which were of interest. So we are looking at brain activity which is happening at the same time, for instance during complex synchronized environment, listening to music, watching movies. We observe that certain brain areas or a large part of the brain starts showing activity which is synchronized, which is correlated. And so if you calculate the correlation between the same brain areas of two subjects, we see that the functions during the perception are enhanced. Yeah, the same was exactly found also during hyperscanning. If you are in social interaction modus at base frequency, these brain activities are synchronized. So the idea is to use neurofeedback to increase those synchronization, or maybe more relevantly to decrease it. What we see here is where we studied brain synchronization in patient and healthy controls, but still without the reinforcement of hyperscanning, but this is a direct social neurofeedback paradigm. But they are looking at between brain synchronization. We see that indeed, particularly the right inferior frontal gyrus, so frontal brain areas start to hypersynchronize in PTSD patient. This is a meta-analytic approach. We can now also pool different groups. Here we pooled two different studies, but this can also be used to study intersubject correlation across several diagnostic groups. And then we see that some groups, this PTSD group, for instance, shows increased synchronization, whereas in schizophrenia we rather see a reduction pattern in this synchronization. So we can try to train this. And again, if we put that in a virtual environment, here we see a live interaction. So this is a multiplayer game, and these two subjects are not, as in, for example, EEG feedback, to look at each other, but they are playing in the same virtual room. And whenever they synchronize the brain activity, for example, now after being reborn, they use very similar activity, and that leads to an increased visibility of the environment and improved possibilities to receive reward. Yeah, and so thereby we are hoping to be able to directly address dysfunctional brain activity in psychological symptoms clusters. Yeah. And the idea is kind of following this total recall model, that by having this reward mechanism, indeed potentially some kind of copying of brain activation pattern is possible, and we are trying to use it in a good way when this method is fully established. I have to acknowledge that this is work from many different people contributing to the MR measurement technology, imaging of different modalities, and the specific neurofeedback application, of which Jana, you just heard. But there are many more people putting their work onto these projects. Thank you, and I hope there will be opportunity to hear some feedback from you.